So now I am very pleased and honored to introduce Dr. Howard Hugh. Dr. Hugh is Professor of Environmental Health, Epidemiology, Global Health and Medicine, and the founding Dean of the Dalla Lana School of Public Health, a faculty of the University of Toronto. Since 1990, Dr. Hugh has led multi-institutional and international teams of scientists, students, and fellows devoted to investigating the environment, nutritional, social, psychosocial, genetic, and epigenetic determinants of chronic disease and impaired child development in population-based studies. His team's work have generated over 300 publications. He has won a number of awards, including the 1999 Progress and Achievement Award from the U.S. National Institute for Environmental Health Studies, and the 2009 Linus Pauling Lifetime Achievement Award, the 2011 Award of Excellence from the American Public Health Association. Please welcome Dr. Howard Hugh.
First, thanks to Richard Fisher for this lovely invitation to address you all. About 15 years ago, I had spent 20 years at the Harvard School of Public Health and the Brigham and Women's Hospital in Boston. And the hospital was holding a fundraiser, as it often does. And the host was very nice. She would introduce each person when they came into the room so that they could mix with various potential donors, etc. And the person before me was introduced as the chief of surgery. And I walked in the room, and I was introduced as an epidemiologist. So a few minutes later, I was standing by the food bar and getting some refreshments, and an elderly gentleman came up to me and said, Dr. Hu, I'm really glad to meet you, and I thought I'd get your advice because a few weeks ago I had a pain in my groin, and I went to my physician, and he diagnosed epididymitis, so I figured I'd check with an epidemiologist about, what I could do. And I guess, we epidemiologists, and I still practice medicine, I'm a clinician, but often we say we don't get respect. But today, in fact, people read the headlines. And so much of what you read about in terms of what gives you the evidence to make choices as a society and as individuals is driven by excellent epidemiology. And that's what I'm here to talk about today. That fantastic talk you just heard from Dr. Gelb was about patients and individual patient decisions and choices that you make as clinicians. But today I'm going to talk to you about populations and some very big decisions that have to be made, particularly with respect to this one subject, fluoride. I've been asked to explicitly state that I do not have any financial interest of a product in my talk or with any companies offering grant monies for this continuing dental and medical education program. Thank you. All right. Let's see if I can get this thing to work. Now, today's talk is about not only fluoride, but also the prenatal environment. And what we're beginning to realize in environmental health, and our research is funded by one of the NIH institutes, the National Institute of Environmental Health Sciences, is that the prenatal environment is much more important to setting us on trajectories of health than we ever imagined. And this is starting to get into the popular discourse in terms of nutrition, in terms of stress, but it's also so important when it comes to pollution. Some of this has been appreciated with disasters like the Minamata disaster. You may remember that, where the industrial pollution in this city in Japan led to marine environment pollution with mercury, biotransformation of that mercury into methylmercury, and the uptake along the food web into the marine species. And the Japanese, of course, are among the highest fish consumers in the world, but it was those compelling images of women who were giving birth to babies who were already neurologically so disturbed that led to the recognition of Minamata disease. Well, that may happen with clinical disease, but now we're recognizing that lots is happening in the subclinical environment, so much so that we have our own societies now trying to study this, like the PP Tox Society, Prenatal Programming and Toxicity. Now, this project, called the Early Life Exposures in Mexico to Environmental Toxicants Project, or ELEMENT, was begun as a multi-institutional collaboration over 25 years ago with many colleagues at many institutions, but primarily with the National Institute of Public Health in Mexico, because we were able to harness the maternity wards of the major perinatology institutes in the city to start following women prior to pregnancy, during each trimester of pregnancy, collect specimens, measurements, and follow the offspring every 6 months so we could develop an entire archived biological data bank and measurements of phenotype to understand what are the prenatal determinants of growth trajectories. This has been funded continuously by NIH since 1993. I'm going to just tell you a little bit about the initial findings, because this gives you a sense of how we can harness a study like this to get at some big issue environmental health subjects. Our first focus was on lead. Why lead? Well, you all know that lead makes kids stupid. We'll talk about that for a moment. But in fact, lead is one of these premier population-wide exposures. It's not just the kids who eat lead paint. In fact, it's everybody in this room has lead in their bodies because of the multitude of pathways through which we've been exposed. One of the biggest sources of exposure was not just paint, and in Mexico, ceramic ware, but this. This is a graph from a chart developed by geochemists, and it depicts atmospheric deposition of lead in ice core samples collected in northern Greenland, where there were no industries, no cars, very few humans. But what you're seeing with these ice core samples are the slow progression of lead that's been deposited from atmospheric pollution right here, and this is when we put lead in gasoline. All of a sudden, this gigantic spike of lead exposure worldwide. The end result is that all of us have some lead in our bodies. Some have more than others. And these are examples of the kinds of studies that have shown us that lead at even these very subclinical levels of exposure makes kids stupid. And this chart on the left is from the Port Pirie study in Australia, where lead exposure and blood levels were measured in women at birth, and the children followed all the way till age 5, and then the exposures were associated with the IQ performance on the McCarthy scales of intelligence after adjusting for all the various covariates. And one of the interesting features of this study, which was conducted in Port Pirie, Australia, a smelter town where lead swirled from the lead smelter around the city, was that the meteorology of that particular city made it so that the high-income kids were exposed as well as the low-income kids, removing socioeconomic status as a major potential confounder of the lead-IQ relationship. And you can see here that as the blood levels rose in these kids, the IQ levels declined. At the time this study was designed and executed, the allowable amount of lead in a child's blood was 40 micrograms per deciliter, which is right here. So way below the federal standard, there was already a dose-related decline in IQ. This is the same kind of analysis we performed in the ELEMENT study, the Mexico children, showing that this decline in IQ occurred even at blood levels, well below 10 micrograms per deciliter, which is the federal standard right at 1991 when this study was performed. And the actual data worldwide now shows that there is no obvious threshold below which we can't see an effect of lead on IQ, making this the premier neurotoxicant that is the subject of so much federal legislation, regulatory efforts, and things that you hear about from the Centers for Disease Control and our public health departments. The reason why we did this study in Mexico, though, related to another issue, which is that, in fact, lead is stored in bones all over your body, and there it resides for decades. It substitutes for the calcium and the hydroxyapatite of bone. Remember that because fluoride has the same action. The worry was that, well, you might learn that you're pregnant and then adopt the most safe lifestyle possible, going to Whole Foods to get your food, which we call whole paycheck because it's so expensive, and eating the right stuff. But because that lead is stored in your bones, and because your bones are literally dissolving themselves to provide calcium for the growing fetus, is it possible that that lead is coming out of bones and still jeopardizing fetal neurodevelopment, even though you're leading a very clean life? And in fact, that's what we chose to study, using some special methodology to measure lead in mom's bones. Why did we have to do that? Because in fact, as this chart shows, this is a chart. When I first saw this chart, I thought it was a chart of the various offices and bureaucracy of the Environmental Protection Agency, but it's not. It's the various human pathways of exposure from all the sources to the human being right here at the bottom. The reality is that you cannot use a questionnaire for anybody in this room to figure out how much lead in your bones. You actually have to figure out how to measure it. So we developed an instrument called a K-X-ray fluorescence instrument. A couple of very bright physicists from MIT developed this. It works very much like a bone densitometer, which is the instrument we use to measure grandma's bone density, right, to see what her risk is for bone fracture. But it measures lead as well as calcium. And this is used in various academic environmental health research centers all over the world now. We built this instrument in Mexico, used it to measure lead in bones of moms, and this is the kind of stuff that we found. Mom's bone lead levels, apart from what her blood lead levels were a major driver of low birth weight in these offspring. This is published in Pediatrics in 2006. And interestingly, when you look at how toxicants may affect development, you can see a lot of it depends on timing and the various stages of fetal development of the various organs and organogenesis through fetal development. Well, that also goes for the brain, because we also found that if you measure plasma lead levels at each stage of each trimester of pregnancy, the first trimester of pregnancy lead levels were far more influential than the second or third trimester lead levels in predicting lower IQ at age 2. That's not great news, because that basically says if you wait until you find out that you're pregnant and then you try to lower your lead exposure, it may already be too late because the first trimester is just about over. That's the kind of research that led to all sorts of different insights on lead exposure and has led to the American College of Obstetricians and Gynecologists to recommend screening for lead exposure for women who may be at risk. We also ran a randomized clinical controlled trial funded by NIH of calcium supplements to reduce the mobilization of lead from the skeleton during pregnancy. And now that's all sort of environmental history. That's all the kinds of exposures that we've been working on over time. But how about fluoride? And why and how did we get engaged on the fluoride side? Well, you of all the people know about the polarization of the world on fluoride and the issues that may be involved. This is, in fact, a really interesting issue for our community because it's really kind of the one side of the public health community trying to figure out how to balance risks and benefits with another side of the public health community, public health dentistry, public health, environmental health. How do we actually solve this issue? Well, in 2007, the National Research Council put out this monograph from an expert panel that was intended to try to look at all the literature that there was at the time about fluoride and its risks. And it was very clear that fluoride, which remains one of the drinking water contaminants regulated by the EPA, is quite widespread because of the water fluoridation policies throughout the country. And it's estimated that in 2000, about 162 million people had artificially fluoridated water in the US. Here's where we're focusing on, because this monograph reviewed the literature and came to the conclusion that, in fact, there were some worrisome animal and human studies that suggested, in fact, that fluoride has neurotoxic effects. But most of the epidemiologic studies were in China. They were what we call ecological studies, where instead of measuring fluoride exposure in individuals, we measured fluoride in water samples of an entire community and then compared neurobehavioral performance of one community versus another. So you're not using individual measures of exposure, you're just correlating fluoride levels in community with IQ. Those are not what we would consider rigorous environmental epidemiologic studies. So our team said, well, we need to look at this much in more depth and see if we can shed some light in this debate. This is a summary table. You can't probably read it, but it was published some years ago that looked at the entire constellation of epidemiologic studies on fluoride at the time. And the conclusion basically was, from my colleagues Anna Choi and Philippe Grandjean, that in fact when they did a systematic review with a meta-analysis of all the studies, they could see a standardized weighted mean difference in IQ between the fluoride exposed and the non-fluoride exposed using biostatistical techniques. So the conclusion was that there may be an adverse effect of high fluoride exposure, but of course we need more and better research. So NIH, in its wisdom, supported us to look at this issue. And why were we in such a good position to do it? Because, as I mentioned, we had all these archived biological samples taken in all these women. And although we hadn't measured fluoride from the beginning, we could go back to these archived samples, particularly of maternal urine taken during the first, second, and third trimester of pregnancy. And it turns out that maternal urine fluoride levels adjusted for urinary creatinine are one of the best biological indicators of fluoride exposure for an individual. So we had plenty of urine samples, and now we're able to, in a compressed time frame, look at prenatal fluoride exposure using these biological markers, and we already had many measurements of cognitive development of these individuals at age 4 and at ages 6 through 12. Now, in Mexico, where we had performed this study, fluoride, instead of being put into water, is put into salt, again for dental health. Why? Well, in Mexico, as you can maybe imagine, the tap water is not quite as safe as it is in the US. So there's many individuals who avoid tap water, and it's not a consistent source of consumption that would enable it to get at the dental public health aims of getting fluoride to everybody. So instead they put it into salt. But because urinary fluoride is a biological marker of what's been absorbed, it gives us a nice indicator of fluoride exposure from whatever source. Now we're able to correlate the early life exposure to fluoride to all these different developmental milestones, beginning with neurobehavioral development. So the subjects for our study were 327 mothers from particular cohorts where we had all these prenatal urinary samples. These are women who are who had attended these 3 Social Security hospitals in Mexico City that primarily serve low to moderate income populations. And these are some of the eligibility criteria from women entering the study, but we used very rigorous protocols for gathering the samples and measuring all of the maternal and the offspring phenotype measures over time. The first analysis we did and published a couple of years ago were the urinary and plasma fluoride levels in these pregnant women. What do they look like? In fact, the mean urinary fluoride levels, creatinine corrected, here were about 0.9 milligrams per liter. That's about twice as high as they were for a national survey of Canadian women, but far lower than they were in the population studies in China. So we're getting exposures that were moderate, not as high in China, but they will be very informative for what risks there may be entailed in women in North America. It turns out that the fluoride exposure markers over each stage of pregnancy have a moderate correlation. In other words, women's diets varied over time, and it's not completely, you can't completely use a single trimester of fluoride exposure to represent what the entire pregnancy of fluoride exposure is. So in our analyses of cognition, we averaged all the fluoride levels over pregnancy. Our study was then published in October this past year. Prenatal fluoride exposure and cognitive outcomes in children of 4 and 6 to 12 years of age. We used maternal creatinine-adjusted urinary fluoride as the biological marker, McCarthy scales of children's abilities to measure general cognitive index, which is a measure of IQ at age 4. The Wechsler abbreviated scales of intelligence to measure full-scale IQ at age 6 to 12, and then a number of statistical techniques to control for any potential confounders. This is just a flow chart giving you a sense of the subject recruitment, follow-up, and retention over time. But basically, this is a study that has been generating key results on environmental risk factors for child development for 25 years. And we have, and our peer reviewers have a lot of confidence in the integrity of the study. Among the things you look for as an epidemiologist is to try to see if there are any subgroup differences between groups of individuals that might account for any differences that you see in your analysis. We did not see that in our study. And then we did these crude analyses looking at how IQ varies across categories of maternal education, maternal IQ, the home environment, and all the other covariates that you have to adjust for in a rigorous epidemiologic study. Now, these results are epidemiologic speak, but let me try to interpret this for you. What we did in this study was take a base model, that is an epidemiologic multivariate analysis that accounted for a number of factors that every study of offspring IQ needs to adjust for, things like maternal IQ, birth weight, maternal age, child age, nutritional factors, etc. And then we sequentially added other factors to see whether it might affect the impact of fluoride on IQ. This is what the base model shows, which is that among 287 mother-offspring pairs, we could see a decline in 3.15 points of IQ for a 0.5 milligram per liter increase in fluoride. That's kind of an interquartile range, going from the middle of the second to the middle of the fourth quartile. A moderate increase in fluoride exposure for an average woman. We also saw for the older children a 2.51 point IQ point decline with that degree of fluoride exposure. And then as we put these models through all these rigorous sensitivity analyses, trying to see if there's any difference in the impact on fluoride exposure, adjusting for the home environment, children's fluoride levels when they were measured later in life, socioeconomic status, lead exposure, mercury exposure, calcium ingestion, the effect of fluoride did not go down. In fact, it seemed to go up. So in terms of the independent influence of maternal fluoride, it's clear that it's separate from what any influence there might be of the children's fluoride exposure. Because in fact, we had children's fluoride exposure, that is, when they are exposed at the time that they had their IQ testing. And the children's IQ that was very modestly associated with their fluoride exposure, but it was much more associated with their prenatal fluoride exposure. The bottom line is that children's IQ, children's fluoride seems to also have an additive effect, but it's not significant and not as influential as prenatal fluoride exposure. This is what the raw data looks like adjusted for various covariates. Here's the maternal urinary fluoride levels. Here are the McCarthy scales of intelligence, the Wechsler abbreviated scales of intelligence, the scatter plot of all the points, and then these multivariate regression lines adjusting for all these different factors. Now this curve is kind of interesting. It's called a smooth curve because it doesn't try to impose a linear relationship between fluoride and IQ. And there's a very, very mild suggestion that there might be a threshold where perhaps fluoride doesn't seem to have effect until a certain level of fluoride exposure, and then the IQ seems to drop. But basically, we don't have enough data to draw any conclusions as to whether there's a safe level of fluoride. Now, when you're in environmental health, because you can't do randomized trials, you can't do a randomized trial of fluoride to see if it affects IQ, just like you can't do a randomized trial of lead exposure or mercury exposure to see if whether lead or mercury affects child development. You have to use not only these observational studies, but the totality of the animal evidence, the tissue evidence, the in vitro experimental evidence to come to conclusions that will then push policy forward. And here the experimental evidence is also worrisome because in fact we can see a neurotoxicity signal in some of the most rigorous research that's been done on fluoride in rats, on both behavioral as well as cognitive outcomes when we look at other rodent studies in mice. And when you put it all together, from our point of view, as we concluded in our study, it's worrisome. And in fact, this is the kind of research that needs to be reproduced in other general population studies, and those kinds of studies are already in progress. We have a collaborator who's working on this very same subject in the maternal infant research on environmental chemicals birth cohort study in Canada, and there are other investigators around the world who are rushing to try to figure out whether this might be true. Some have questioned whether those urinary fluoride levels in pregnant women are relevant to making general population conclusions, because physiology changes during pregnancy, and perhaps comparing urinary fluoride levels in pregnant women with urinary fluoride levels in the general population is comparing apples with oranges. Well, there are some general population sample studies of urinary fluoride. Here are two of them, one reported in Israel, one in Poland, where the urinary fluoride levels in these general population samples are very similar to ours. But in fact, the MIREC study that I just mentioned in Canada just finished its analyses of urinary fluoride levels. It hasn't been reported in a journal yet, but their average level is about 0.6 milligrams per liter, which is even closer to our average urinary fluoride level of 0.9. So our urinary fluoride levels are clearly meaningful to what happens in North America. Now, once we published a study in October, the media picked it up, even though it was a classic Trump-driven news week where there's lots of stuff going on. We felt gratified that most of the speculation was regarding the interpretation of the results for policy rather than speculation about the integrity of the science. The American Dental Association did review the research and came out with their own interpretation. And I just highlighted their what I guess could be construed as their conclusion, which is that because it's unknown how the subjects of the study ingested fluoride, whether through salt or water, no conclusions could be drawn regarding the effects of community water fluoridation in the U.S. I think that's a red herring, frankly, because as I said, we used biological markers of fluoride. It doesn't matter how you're exposed to fluoride, whether it's water or salt, it's still going to give you the same biological marker. I guess, we'll see how this plays as the policy wheels turn going into the future. But perhaps most importantly, some have questioned, well, if you're losing a few points of IQ, what's the big deal? Well, first of all, would you like your child to lose 3 to 5 points of IQ? I don't think so. But perhaps even more importantly, there's the population-wide angle. Because in fact, when you're shifting the entire population over 3 to 5 points, you're in particular affecting the extremes. You're pushing people off the cliff into the mentally retarded zone. You're really robbing society of those who are at the top end and who are contributing a lot to the knowledge economy. But the other aspect is that this is an epidemiologic study. We average what the result is in the population. So if you're losing 3 points of IQ as an average in a population, no doubt there's some kids who are losing only 1 and some kids who are losing 5 or 6. The second point as an epidemiologist that has to be expressed is that this is based on 3 measures of fluoride during all of pregnancy and then averaging. How well do you think that represents a person's entire biologically relevant fluoride exposure during pregnancy? This is probably what we call measurement error. Measurement error typically reduces the estimate of what the impact is on the outcome. So in fact, that 3 points of IQ might actually be 4 or 5. The reality is that we have to do a lot more research to really understand these kinds of effects. And until that happens, we need to already start thinking about what the policy implications might be. There's also plenty of research that has looked at what IQ loss might mean for economic productivity. You may be familiar with some of that. And I guess when you think about how environment may play related to all the other things that are known to affect child intelligence, it really bears thinking about how we might compare this from a relative scale. This is a publication that my colleague David Bellinger made in 2012, in which he used the estimated full-scale IQ point loss associated with different risk factors in a population of 25 million children and extrapolated all the different what's known about risk factors for IQ. And certainly preterm birth is a biggie. It's right there. 34 million points of full-scale IQ loss. And some of these other things would seem plenty relevant. But right down here is lead. 22 million points of IQ based on what we know about lead exposure in the United States today. Organophosphates at 16 million. Methylmercury at 284,000. These are not small effects. And we have to now start persuading our policymakers that it makes sense to think about pollution, whether it's intentional or whether it's through the other activities of human industry. Now, we are used to doing controversial research, so it was not surprising when we received a Freedom of Information Act for our research findings and our data from the Fluoride Action Network and from folks who are on the other side of the spectrum in the who do epidemiology for the American Dental Association. But our basic position is keep our head down, do the science, we'll generate the best evidence, and let the policymakers figure out how to take the results and do the right thing in our communities. These are some of the things that are ongoing that we plan on publishing over the next few years, looking at earlier life cognitive outcomes, the Bayley Scales of Intelligence at age 2, looking at behavioral disorders like attention deficit hyperactivity disorder, autism-like behaviors, looking at potential interactions with other pollutants that we've measured over time. We also have a lot of genetics in our research where we've used a candidate gene polymorphism strategy to genotype our mothers and our offspring to look at genes that are high candidates for adjusting an individual's susceptibility to environmental toxicants. And then there's this other interesting biological marker that it turns out, and this will be interesting to you, that the shed primary deciduous teeth are beautiful media to actually use these state-of-the-art methods, here this laser absorption microscopic capture methods, to interrogate the dentine levels. And we can actually figure out what the prenatal toxicant exposures are at 2-week intervals using that biological marker. So we have been busy collecting the shed deciduous teeth for all of the children in the element study, and we'll be using this biological marker to good effect for our research going forward. As I mentioned, Christine Till is working on the MIRA cohort research with a very similar set of aims as to our own, and we'll be discussing a lot of this research at one of the biggest meetings of public health, the International Society for Environmental Epidemiology annual meeting in Ottawa this August, where there's a whole symposium on the policy implications and how we examine dental benefits, neurobehavioral risks, and make policy recommendations. Anyway, with a birth cohort like this, we can examine a whole number of other toxicants. Here's just a few that this research project has either examined over time or that we're examining going forward, and some of the other outcomes that we've looked at, blood pressure, sexual maturation, obesity, and the metabolic syndrome. But the bottom line is that this is the kind of research that we hope will be persuasive to the policymakers because we use the best, most rigorous possible methods to measure both the exposure and the outcomes and the covariates. And the rest is up to you and the policymakers to make the right decisions both on the individual level and at the governmental and a societal level. And this is just a little bit of environmental health humor to entertain you all. Thank you very much.